So I'm so excited today to talk to Dr. Helen Dooley. She is a associate professor at the University of Maryland in microbiology and immunology and a shark expert. So the motivation behind this podcast is we work with a lot of customers who are interested in developing therapeutics and reagents that are built on single domain antibodies. And traditionally, we've used llamas and alpacas, but of course, single domain antibodies can be generated also from shark IgNARs. So we're excited to hear about, how to use those in our research, and would love to hear a little bit of background from you, Dr. Dooley.
Mhm. Yeah, so we're interested in the shark immune system for two reasons. One, we're interested in understanding just basically how immune systems evolve and change. It's tricky to get money for that at the moment. So the other kind of line of research that we use is to look at the single domain antibodies that we can generate from cartilaginous fishes, so sharks, skates, and rays. And basically, sharks have three different antibody isotypes. So they have a conventional IgM that's just like our IgM, but they have that in a monomeric and a pentameric form. They have an ortholog of IgD, which is called IgW. And then they have this really cool heavy chain only antibody called IgNAR. NAR initially for new antigen receptor, because we weren't even sure that it was an antibody when we kind of first found it. And unlike the camelid antibodies, which I guess people are more used to hearing about, these heavy chain antibodies in sharks have kind of arisen by convergent evolution. So even though they kind of look the same structurally, they're very different. So in the camelids, you have an IgG that's basically mutated away from the ability to be able to bind to light chains. So they're much more like conventional antibodies that we see, heavy light chain antibodies. So for IgNAR, our understanding of it is that actually a single domain that was present in T cell receptors, this weird T cell receptor type that sharks have, managed to invade an Ig cluster. So this domain has never, ever interacted with a light chain. Structurally, it's kind of a cross between a light chain and a T cell receptor. And it's basically there is the binding region for the constant domains that kind of came from an IgW cluster. So even though they have the same sort of structure, they arose very differently.
And are they in the same locus? So camelids, all the IGs are in the same locus, and so you just sort of pick a different germline, and then it's just like a class switch to get to the heavy chain only. Is it kind of co-located with the IgM and IgW?
So we think so, because we see sometimes we think all the IGs are clustered together in the genome, and we're not absolutely sure because we haven't sequenced the entire genome of any shark yet, and those bits are really difficult to figure out because there's a lot of repetitive elements in there and a lot of duplication. But into the Shark 101, shark antibodies aren't in translocon, like human antibodies are. So you don't have lots of V's, lots of D's and J's, then with your constant regions of the different isotypes. So instead, you have kind of the V, the D, and the J that you need to make up the V region, and the constant regions for one isotype, and then lots of copies of that, what we call cluster. So they don't actually isotype switch. So we think shark B cells basically get programmed to express one isotype or another right at the very beginning during rearrangement, and then kind of are locked into that. So you never switch from kind of IgM to IgNAR. The B cell is always an IgNAR B cell as far as we know.
And you said it's kind of a cross between a light chain and a TCR. What makes you
Mm-hmm.
say it's a light chain as opposed to a heavy chain?
Um, Kind of structurally, it looks more, it's kind of got more similarity to an Ig light chain. We don't exactly know what it is, to be fair.
And do we see the same kind of CDR, like antigen binding regions that are hypermutated. There's three of them, and that's kind of
Nope. So cool stuff with the what we call the VNARs, the binding domains. So to generate a functional VNAR, the shark has to rearrange a V segment, three D segments, and a J. So your 3Ds give you CDR3, just like in other antibodies. But because you're rearranging 3Ds in there, you've got four rearrangement events, you've got trimming, you've got NMP addition, you've got all the cool stuff going on. That means in terms of sequence length and sequence diversity of CDR3, they have this huge CDR3 repertoire. So we've seen everything from, a 5 amino acid CDR3 right the way to 28 or 29 amino acid CDR3s. They also encode sometimes, if you use the D in the preferred reading frame, they have cysteines encoded in them. So you can fold the CDR3 in different ways. And then for the rest of the domain, there's actually a truncation in framework 2. So they have a conventional CDR1, don't have a CDR2, have a conventional CDR3, but then like T cell receptors, they have another loop, which we call hypervariable 4, which kind of sits behind CDR1. And then because of the truncation, instead of CDR2, they basically have a loop that runs like a belt around the, I guess it would be the face of an Ig that would normally interact with either light chain or heavy chain. It would be the pairing interface. It kind of runs around the center of that, and we call that hypervariable loop 2. And all of those loops can be mutated. Sharks actually have one of the highest rates of somatic hypermutation there is. So we get these really diverse primary repertoires that then get shotgunned right the way through with a lot of mutation. We quite often find what we call families of clones, where we have, one CDR3 rearrangement event, and then we'll get 30 or 40 different clones that have been differentially mutated. So we get to do some really cool studies with that, looking at how mutation affects affinity, where in the molecule mutations either improve or reduce affinity, or how they're used to interact with different antigens. It's pretty cool.
so is it sort of like rabbits where they kind of, when they're young, they kind of create their primary repertoire and then everything is sort of mutating from there, as opposed to like other species where there's a lot of turnover and so you see a lot of new naive sequences throughout the life.
Yeah, so rabbits have a very kind of limited primary repertoire, just because of the way that their immune system works. We're not seeing that in sharks. Our kind of theory is we've recently kind of characterized the shark equivalent of a germinal center. And we think that the selective pressures in the sharks are maybe not as great as you would see in a human. So you can carry through a lot more diversity in your repertoire post-mutation, which makes sense, I guess, if you're an animal that's immune system doesn't work quite as speedy as humans, and you want to generate as much diversity as you can in your repertoire in case a virus or something is mutating away from the response that you've produced.
But there's no concern about self-reactivity because I feel like that's often what kind of like, drives
Uh-huh.
the reduction of the immune response is you accidentally end up binding to yourself. You don't see
Yes.
that.
I mean, not so far. Honestly, how some of this works is a complete mystery to us. I will say that kind of the physiology of sharks is different. So they do have a lot more salt and a lot more urea in their system. So their antibody is probably not quite as sticky as ours, like in vivo. Um, and they must have, every vertebrate that we've seen that has B cells has some sort of selection mechanism to try and get around autoreactivity. And, we presume, although we don't know how, that that's also working in sharks.
So you mentioned that the physiology in which the antibodies are being matured is different.
Mhm.
do you see differences then in terms of kind of stability or, um, well, you said structure already, but do they work kind of the same in the lab or in the body?
So we know that because of the physiology of the sharks, a lot of their proteins are a lot more stable than our equivalent of that protein, because they're operating in these high urea, high salt environments. And actually, the way we think about it is when our ancestors first came out on land and their physiology changed, probably our proteins had to become a little looser and a little more flexible so that they could work properly in this new environment. So we do see that they're more stable. Definitely the VNARs are pretty resistant to denaturation in things like urea, obviously, but also temperature stable. So we can take them up to kind of 60 degrees usually before they start to unfold. And then like the camelid VHHs, what's really nice is then if we reduce the temperature, they actually fold back to native configuration instead of crashing out of solution. So it seems to be, they seem to be very soluble and very able to kind of recapitulate the original structure once you've denatured them.
Is there any risk of aggregation? I mean, are they kind of, do you see secreted IgNARs in the same density that we see sort of IgGs in the human body?
So sharks have really, really high levels of Ig in their bloodstream. So it's mainly IgM. So it can be up to kind of, I don't even know, like 50% of the blood plasma is IgM. IgNAR is about 10% of that. We don't see proteins crashing out. If we just express the VNARs on their own, we can take them up to silly concentrations in the fridge before we start to see them dropping out of solution. And then usually what we do is we just stick them in the 37-degree water bath for a bit and they go back into solution and they work perfectly fine. But we're taking them up beyond the mgs per mil range for them to start crashing out. They're really, really soluble, nice to work with.
Yeah. And I guess one last, you know, immunobiology geek question. Can you
Mhm.
pull out the CDR3 like the cow ultra longs? Those 28 amino acid CDR3s, are they sufficient for binding on their own or they really need the whole scaffold?
So we have instances where, say, if the CDR3 forms a protruding loop, like a finger on the VNR domain, and then enters into a cavity or something on the protein, then we could, say, take the CDR3 and put it on a different scaffold and use it that way. But a lot of the time, we're also looking at interactions from other parts of the protein. I think one of the things we're probably going to get to is things like affinities. So these guys tend to vary about the same amount of surface area and interaction as a regular conventional antibody. So to do that, they have to use a lot of the actual VNAR surface to be able to get that interaction. One of the ways they do it is by inserting some of these loops into clefts or pits on the protein to increase that surface area.
Interesting. So then I guess talking about epitopes in camelids, because they have conventional antibodies and heavy chain only antibodies, there are certain epitopes that are preferred by
Mhm.
conventional versus heavy chain only. Do you see that same kind of preference? Certain epitopes are just better targeted by the IgNAR versus the IgM.
Yeah, absolutely. So, like I said, we see a lot of VNARs that bind where they will insert CDR3 or say the tip of CDR1 or something into a pit or a cleft on the surface of the protein. The other cool thing about them is, like I said, because they've never interacted with a partner, um, whereas conventional antibodies and VHHs tend to work through that top interface, which would have been, the binding site when the VL was there. They tend to come in through the top and interact mainly with kind of CDR3 and CDR1. Because VNARs have never had that and they have this hypervariable loop too, they can actually come on kind of side on, like a ladybug on a stem, and use the CDR3. Sometimes the CDR3 is folded over and that CDR HV2 loop to kind of bind to flatter surfaces. And that's a mode of binding really that no other like traditional antibody out there or VHH antibody can achieve. So what we've seen, even the massive amount of data that's out there at the minute with the SARS-CoV-2 spike and all the epitopes being mapped on that, we actually have now a range of VNARs that bind to completely different epitopes to the VHHs and to the conventional antibodies that are out there just because they have this different mode of binding.
Interesting. So when you, I guess, can you walk us through what kind of an immunization campaign for a new IgNAR would look like? What species, how long does it take, all that stuff?
Yeah, so there are a couple of teams out there now using different species. We kind of pioneered this technology in nurse shark, and that's the sharks that I use. I'm very lucky as I work for University of Maryland, Baltimore, but I'm placed in the Marine Center here, so the Institute of Marine and Environmental Technology, which means we have an awesome aquarium downstairs. And I basically have two big tanks of sharks, which I fully believe makes me a supervillain. So we would, if we get a new target, we kind of, talk to the collaborator or whatever about the target. The first thing we do is go into our database and we'll do a search, like a blast search of our shark protein database to see if there's a protein with any sort of homology to that protein. And if so, what the homology is. And quite often, people will come to us because they have their favorite human or mouse protein and the antibody they've got isn't very good, or it's an IgM antibody and it's sticky and it binds to other things. And they've tried immunizing mice, but you have tolerance because the proteins are so similar and they just can't get a good antibody. If we look at sharks, then you're 500 million years separated from humans. And generally, if we look at the same proteins that might be highly conserved between mouse and human, or even camelid and human, we're usually looking at less than 50% identity in sharks, unless it's something that's super conserved, like a transcription factor. But things like receptors on the surface of proteins, things like that tend to evolve pretty fast. So generally, we're operating in the 50 to 30% identity range, which means that, we don't have to break tolerance. These animals are not tolerant. This is not a shark protein. So then we would basically, we go downstairs. We normally immunize at least 2 animals per target. And we'll take a pre-bleed for our baseline level. And then we immunize the animal. And we kind of use, first of all, we would immunize subcutaneously. So into the fin. So just like us getting our flu shot in our arm, we kind of inject the protein with adjuvant into the shark fin. And then dependent on the protein that we're working on or the molecule that we're working on, then we would boost at about monthly intervals, either subcutaneously again or IV with no adjuvant. We take blood samples about 2 weeks after, and then we would basically just run binding ELISAs to see when the antigen-specific IgNAR levels come up. When we first did this, it would probably take, say, 4 to 6 months. So 4 to 6 immunizations to get a good response. We've honed that a little bit now, and dependent on the target and how we kind of display it to the immune system, we've definitely got that down, 4 months for some targets. We're working on a target at the minute where the animals have only had a prime and a boost, and we're already seeing really nice titers because it's a nice immunogenic protein that we've presented. We present a lot of things now on nanoparticles to kind of mimic more like the pathogens maybe that the shark immune system would see. So in that case, I think we're going to have the sharks are going to be ready to build libraries from in 8 weeks maybe, which is really nice. I feel is pretty comparable to a lot of the systems that people are using. Once we have high titers, basically we take a blood sample from those animals, isolate PBLs, and then we can build large VNAR libraries, in a couple of days. So it's only 3 primers. We don't have this worry with the camelids where you're trying to differentiate the conventional antibodies from the VHH antibodies. IgNARs are so different to all the other antibodies in shark. We have pretty specific primers. It's, one PCR. If I'm being really good, we can go from, blood in the animal to a 10 to the 10 library in a day or two. And then, we'll just then start panning, dependent on what the person is. We tend to do phage display, but we can tweak parameters. We've worked with collaborators that have built yeast display libraries to use that approach. So they seem to be pretty amenable, at least to the techniques that we're using generally to get binders.
Hmm. Are there tools kind of readily available in terms of primers, secondaries, or is everybody kind of has to let you glean the literature and roll your own?
Yeah, so this is build it from the ground up. So I mean, we have protocols out there for nurse shark that are pretty detailed and have all the primer sequences in. They've been out for a long time. With other shark species, I think people are like, oh, a shark is a shark, but a lot of the sharks, they can be over 250 million years distant. So you can't presume if you've got, VNAR specific primers for one species that they're just going to carry over into another species. You kind of have to do some sequencing and figure out what your primers are. That's the fun bit.
Is there concern about hypervariability in framework one that would mess up any primer? I mean, I guess, do you worry about mutations in the framework one where your primer might sit?
No, so they have a little, it's about 6, 7 amino acids right at the beginning that's really highly conserved. So we're lucky that way. And then the J is really highly conserved as well. So we have 2 primers at the back end just to catch. There's, I think, 2 amino acid differences in our primers to catch that. But it doesn't actually seem to make that much difference in terms of binders. So we seem to capture everything with the primers that we have. It's the advantage of nurse sharks only have like 3 IgNAR clusters, and only 2 of them are expressed in adults. So we have, the sequences of those worked out pretty well.
And you can get enough diversity from a bleed because you mentioned that 10% of the B cells are maybe, I mean, yeah, 10% of the B cells are producing IgNAR versus,
5% maybe.
okay.
Yeah.
But you still get enough diversity from the bleeds.
Yeah. So again, we're not working with mice, we're working with animals
Right.
that are between 3 and 6 foot. So we can regularly take 10 mil blood samples. From some of the really big guys, we can take more than that. That is more than enough
Hmm.
RNA that we would ever need for anything, I think.
Yeah. And do you always run an immunization campaign or have you built kind of like naive libraries that you would use?
So we have built naive libraries before. I can, I'm very fortunate in that I have facilities to keep animals, and I've worked with sharks for over 25 years and, kind of know what we're doing and how to keep them healthy and happy, and just how to handle them. We did a while back kind of try and build naive libraries because obviously the advantage there is you don't have to immunize and you don't have to use that time and you don't have to have animals available. We were just finding that the antibodies that we were getting back were much lower affinity and tended to be slightly cross-reactive, which is, if that's all you have is a naive library, I can see then that you get your leads and then you can mature them out and you can do things to them later. It is much easier for me to let the sharks do the work. And then I just build a library and we can have binders within, a week or two. Where it's kind of much less labor intensive for my team in the lab because the sharks have done all the mutation and the selection for us.
Hmm. Have you had success with co-immunizing so you can kind of be running multiple campaigns at the same time and then just pan against the
Yeah, so we've gone up to, I I think 4 or 6 targets at once is our main one. Part of it is picking your targets carefully. We have seen, so in one of the campaigns where we did 4 targets at once, we got very good responses to 2 of the proteins, reasonably good to one of them, and then one of them we just didn't get much of a response to. And for that campaign, that was a while ago, but we've just decided we're just going to boost the animal now with the one protein that didn't work to see if we can pull back a memory response just against that protein. So the other cool thing is we keep these animals for a long time. Some of the animals that I have in now, we've had, for almost 8 years now. So we can go back, as well as doing foundational studies, understanding what immune memory looks like in them. We get a second shot, maybe at things that didn't work so well the first time.
We find also because llamas and alpacas are outbred, two different animals can have very different
Mm-hmm.
responses to each target. Do you see that also in your sharks?
Absolutely, yep. We do have certain targets that just don't work very well, no matter what we do. A very small percentage of the targets. If a target works well, it tends to work well. But we always, I said we always try and immunize at least 2 animals, just in case something strange happens. Sometimes an animal won't respond. Sometimes it doesn't respond very well. It also gives us kind of 2 bites of the cherry. So we get to build 2 libraries and we can pan them. And just because of what you said, these animals are outbred animals, they're creating different diversities against that target. We try and get, increase the number of leads that we get against the target just by using multiple animals.
And you mentioned that for cases that don't work, is there anything common about those targets or random?
Um, I think so. We couldn't find it in our database, and I think maybe it's more conserved in shark than we thought it was, or maybe it's just a certain fold that the immune system doesn't like to respond against because it tends to be kind of a self protein fold. We have another one that's really small and really soluble that we're thinking we just need to link it up to something. I don't think there's many T cell epitopes there that are just getting carried.
Are you looking for IgM resp like because oftentimes for camelids, if we don't get a good heavy chain, it's because it's just all conventional.
Yep, and we've definitely seen that. So the small soluble target I was talking about. Oh no, it wasn't. No, it was the other one. We got a really strong IgM response against it, so it seemed to have just locked in to this response early on.
And then I guess you mentioned affinity. I guess are you seeing kind of typical affinities out of these when you run kind of the
Yeah,
standard campaign?
yep. So just for a standard campaign, say 2, 3 rounds on the same antigen, we will get, I mean, everything is either nanomolar or subnanomolar as a single domain. If we then, make those as human FC fusions or something like that, we're seeing really, really nice avidity effects from that. And we get antibodies that literally will put them on the BLI and you don't see them come off. They just stick there, which is really nice.
So one of the things a lot of people do with VHHs is daisy chain them and make them modular and bispecific. Are you seeing the same kind of efforts happening with VNARs?
Yeah, I mean, I think one of the nice things about working with kind of any sort of single domain binder is that they're so easily engineerable and you can basically engineer them into whatever format best suits your need. So we have everything from just the single domain on its own, which 12 to 15 kilodaltons, right the way through to we now have constructs that kind of mimic IgMs, and we have IgG type formats, and we have trimers, and we have beads on string, and we have you can play about with them and kind of do more or less whatever you want within reason.
I guess so now that you've been working with VNARs for, you said 20 years?
A long time, yeah.
Yeah. I guess where do you see them having the biggest impact?
Biggest impact. So I mean, for me, I think the fact that we're seeing them bind to very different epitopes to other antibodies, to kind of conventional antibodies and VHHs. So we had a project a while ago in collaboration with Jamie Spangler at Hopkins, where we basically took a therapeutic antibody that's in the clinic and kind of neo-functionalized it by adding VNARs onto it that bound to very different epitopes, and kind of see what we could do in terms of if you're binding more than one epitope on a target protein, what differences you could bring about with that. So I think there's room for it in that if you have, even if you have a conventional antibody, but you need something that's going to find a different epitope on that protein. I think there's room for them, like the camelid VHHs, maybe as kind of enzyme inhibitors and things like that, because they do like to bury themselves. We've been doing a lot of work on viruses and just using them to kind of map the epitope space on viral proteins and look for other points of vulnerability. Whether then that's a VNAR that's used or a small molecule or something like that. But basically, are there different spaces on this protein that we wouldn't see with a conventional antibody?
So then you mentioned one of the benefits of going with shark is that there's 500 million years of evolutionary distance
Mm-hmm.
with human. So then I guess once you get your IgNAR out of the shark, then you have $500 million of difference with human, the
Uh-huh.
human body.
Yep.
I guess how close are we to kind of bridging that gap and having the VNAR or IgNAR be a therapeutic that you could put into a person?
Yeah, so I mean that's a really good question. I mean the big, I know it comes up all of the time, every meeting I'm in, every grant that I put in is immunogenicity, and how we're going to humanize these things. So way at the beginning there were efforts to humanize, but I feel at that point maybe our hands were tied a little bit by not having a huge amount of sequences for VNRs, and certainly not having many structures. I think there were only maybe 3 or 4 structures of VNRs out there. Um, Part of the work we've been doing for the last, kind of 10 years is to build up a huge database of sequences and of VNARs structures with their antigens to understand how these things are actually interacting. I think resources like that will make it much easier to humanize should we need to do that. But one of the things, we haven't done any long-term studies yet, but we've done a lot of short-term animal model studies, and we've never seen any sort of immune response or anything bad going on with these molecules. So the general sense that I have is that maybe they're not very immunogenic. They're
Hmm.
quite small. They don't have a lot of T cell epitopes. They're really soluble. Um, and I think maybe they're flying under radar. And what I'm trying to get is money to actually run a side-by-side comparison. So if you take these molecules and you do immunogenicity studies on them in their natural form, and then maybe a couple of different humanized forms, whether that makes any difference, whether it makes things worse. I mean, we don't know that humanization, like you could humanize these things, they become less soluble, they actually become more immunogenic.
Hmm.
So I think that's something we really need to look very carefully at. But I think just as we acquire more and more information about them, it's going to become a lot easier than it was.
Yeah, and I think the humanization assays become a little bit easier to
Mm-hmm.
implement early on. Not,
Yeah,
it also will
yeah.
be easier. So I guess everyone this year is talking about AI generated antibodies. There have been AI generated VHHs.
Uh-huh.
Do you think that AI generated VNARs are on the horizon?
I mean, I don't think anytime soon. Again, just basically there isn't enough information out there to feed in to get something sensible out at the other end. I certainly still look at new structures and I'm like, wow, okay, I never would have predicted that interaction, or I never would have. So yeah, I think eventually we'll get there, but I think at the moment, like a lot of things, you need enough information in there to actually, be able to interpret and do something with. And I don't think that we're quite at that point yet. Would be very cool. As I said to somebody, I could just, go home and just just let my computer run and it would be great.
So you mentioned 10 years ago there were only three or four structures, but then now it's become kind of a routine part of your campaigns. So
Mhm.
what are we looking at in terms of how many solved structures are there?
I honestly don't know how many are in the database, but I know somebody told me the other day that we have more in our database than that in kind of the protein crystallography database at the moment. So we're actually working on a paper where we can get more of these kind of structures out into the world. Um,
What is your
but
resource for where can people find these structures then if they're not in PDB?
They can't at the moment, but that's why we're
Oh,
working on
okay.
trying to,
Okay, got
yeah,
it.
that's why we're trying to
Application
work on
pending.
get a paper, yeah, a paper out.
Got it.
Hopefully sometime this year. Um, yeah, it's one of those things because we've just started now, kind of we work with partners that do a lot of cryo EM, so we just generally get structures for things. Um, and it suddenly occurred to us that we actually have this massive resource that we need to get out there. So that's hopefully going to happen soon.
Yeah, especially with that kind of diversity of folds and diversity of sequences, that would be super interesting.
Hmm. Hmm.
Is there a place where people can find the like repertoires of shark? Are they easily available?
There is some data online, um, not a huge amount. I never try and think that one system is better than another. I think what it is that we want as many tools available to us. And we just pick the tool that happens to fit the problem. So, whether that be a conventional antibody or a VHH or hopefully at some point VNARs, that people will be able to make those choices.
Hmm. Well, they seem just different enough that you're getting really interesting antibodies out of it. So that's really interesting.
Hmm. Hmm.
Yeah.
I always love it when we get something and it's like, oh, especially the SARS-CoV-2 work, just because there are so many antibodies out there, and then we're like, oh, this is a brand new epitope. We've never seen this before. Like, what's going on? Or this is a brand new mechanism for inhibition. Like, what's going on with that?
Well, thank you so much for your time.
Oh, my
Um,
pleasure.
yeah, and have a great rest of your day.
Thank you very much.